evening. My name is James. I'm the house manager here tonight. On behalf of the staff at Town Hall Seattle, it's my pleasure to welcome you to tonight's presentation featuring cutting-edge research by UW students Risa Takenaka, Lucy Bowser, and Elizabeth Bonner. This is the second of 5 events in the UW Engaged Science series, and we're so pleased to be able to once again offer this platform for these students and their important and engaging work. As we get underway, I would like to acknowledge our institution stands on the unceded traditional territory of the Coast Salish people, particularly the Duwamish. We thank them for our continuing use of the natural resources of their ancestral homeland. We're so glad to have you all tonight. Town Hall is adding new events and podcasts every day. Visit our website to view our calendar of upcoming events and sign up for our e-newsletter to receive updates as future events are added this season. Town Hall's work is made possible through your support and the support of our sponsors. As part of our Arno G. Motulsky Science Lecture Series, this event is supported by Microsoft. After the 3 presentations, we'll do a quick scene change. The podium will move, the stools will slide over for the Q&A. We ask that if you have a question, come up to the microphone so we get a good recording of it. And also during that scene change, you're welcome to head out to the cafe and get a coffee or a drink. All right, now please welcome Nicole Gregorio.
All right. Hi, everyone. Thank you so much for coming tonight. Last winter, I was very lucky to get to be the instructor for the Engaged Science Communication course. This course is run by a group of graduate students at UW. We call ourselves Engage, and we seek to prepare a group of really excellent UW graduate students with the skills that they need to communicate about their work with the general audience. So Engage hopes to improve science communication education at the University of Washington, and we really envision a future where scientists more effectively and freely communicate about their work with the public. Tonight we have 3 incredible students from our course here to tell you about their graduate work. Their talks represent real research happening at the University of Washington right now, so we hope you enjoy this unique opportunity to get a behind-the-scenes look at that. So without further ado, I'll introduce our first speaker, Risa Takenaka, who is a graduate student in the Molecular and Cellular Biology program at the University of Washington and the Fred Hutchinson Cancer Center. As an ecologist turned molecular biologist, Risa is interested in understanding how evolutionary pressures have affected and continue to affect living things from fruit flies to humans at the genomic level. So please welcome Risa.
Thanks, Nicole. So my talk is called The Curious Incident of the Fly in the Market, a nod to Mark Haddon's novel. So I'm going to started the timer, but not the talk. So. Let me do this, and there we go. Oh, the arrow isn't working. Technical difficulties. There we go. So I'm going to start by taking you back to 1967 at a fruit market in Rome. It's a beautiful sunny day, and people are milling about, buying their produce, and people see this one gentleman who just is very out of place. So among the shoppers of produce, here's this American gentleman just carrying this huge insect net, going like this, trying to catch insects, and other people are like, we're just here to shop for fruit. What are you doing? And people are like, oh, maybe, he's catching butterflies, but no, he's catching flies. And people also notice his companion, another American gentleman who's talking to people in fluent Italian and saying, don't worry, he's my friend. He's a little out there, but he's harmless, and I'm just his chaperone to make sure things don't get out of hand. And these people are actually scientists from the US. So we had Dan Lindsley and Larry Sandler, and Larry at the time was a professor in the genetics department here at the University of Washington in Seattle. So the story that I'm going to tell you is a detective story, and our first detective is Larry, and his insect of interest is called the Drosophila melanogaster. Its aliases are fruit fly or vinegar fly, or among the people who work with them like Larry, just D-mel for short. So this story is just one of the many volumes in this metaphorical library of biology. So science and biology is made up of many different, distinct, but related stories. So here are just some of my favorite stories, like Charles Darwin's work on the finches in the Galapagos to study the theory of evolution by natural selection, Gregor Mendel's work on peas to study the rules of inheritance, Rosalind Franklin and DNA structure, and more recently, Jennifer Doudna and Emmanuel Charpentier's work on CRISPR-Cas9. And of course, very useful things like the COVID vaccine, things that enable us to gather here today, things that weren't possible 3 years ago. So today's story, of course, focuses on these Drosophilists. So why fruit flies? Well, poets before scientists knew of the importance of fruit flies to humans. So poets like William Blake back in the 18th century liked to use fruit flies as a model for confronting with their own mortality. And their ephemeral life made us confront the ephemeral nature of our human lives. And they weren't wrong, because fruit flies have a lot to teach us. There's been 6 different Nobel Prizes that have focused on fruit flies as its study subjects, and things like genetics and more recently understanding of circadian rhythms come from fruit flies, and they're also a great model for understanding different human diseases like cancer, diabetes, and even Alzheimer's. And the other reason that fruit flies are great is that they're really convenient to keep in labs. So they can essentially spend their entire life cycle in this small bottle that has little food at the bottom that the larvae can eat, and then they can grow up and become adult flies within more or less 10 days in the lab. So going back to Dan and Larry, what they were doing that day in Rome was looking for flies that were less fertile compared to their friends. So before I tell you about the results of Dan and Larry's work, I'm going to take you on a little bit of a microscopic detour. So if you take this fly and zoom in on the cell, which is the basic unit of life in both fruit flies and humans, you have the cell, and even smaller than that, contained within that is the nucleus. And this is the unit that contains a lot of the genetic information that gets passed on from parent to offspring. If you zoom in closer, you see this thing called a chromosome, and keep zooming in, you see the nucleosome. The names are not as important as appreciating the fact that you can go really, really small and in detail. And the thing that part of what makes up the nucleosome is the DNA. And the DNA wraps around these things called histones, and I'm going to come back to that in a minute. But for now, I want us to take a deeper dive into DNA. So in humans and also in fruit flies, we have 2 copies of DNA, one from the maternal parent and one from the paternal parent. And this DNA contains genes, and genes are these discrete units that have specific functions. So for example, one gene might tell us about our susceptibility to certain diseases. Another set of genes might determine how much pigment to put in our hair, for example. And genes are important because they act as the blueprint for proteins. So the genes contain the instructions, but it's really these proteins that are the workhorses that go out to their bodies and do the functions that the gene is meant to perform. So like all things in life, things change, and it's no exception with genes. So on the left, we have what we call the wild-type genes. You can think of them as the dominant gene in the population, or you can think of them as the typical, unchanged form of the gene. And when genes undergo change, we call that a mutation. And when you hear the word mutation, you might think of like X-Men, or like really bad or scary, but actually in the scientific context, it just is a very neutral word meaning change, and the outcomes can be good, neutral, or bad. And in fact, most mutations are neutral, and we also call them silent, because there might be a change in the gene, but it's so small that the resulting protein just looks like it's wild type. But other times, it's changed so much that no protein can get made, and obviously that's not great, so that's bad. Or maybe a protein gets made, but it's a different shape, and it can't perform its function. But you can also get cases where you get a different protein, but maybe this new version is actually an improvement upon the wild type for the specific function. So different kinds of mutations and different sorts of consequences. So going back to the macroscopic world in history, what Dan and Larry were doing that day was essentially hunting for mutant flies that are less fertile compared to their wild-type counterparts. So they collected all these flies in Rome over the summer in different fruit markets and in different wineries, and they brought them back to Larry's lab in Seattle in the fall. And one of the important questions they wanted to ask was, what is the mutation that is responsible for this reduction in fertility? And Larry discovered that it's actually this gene called ABO. So in wild-type flies, all flies have this gene called ABO that makes this ABO protein, and that results in typical or wild-type fertility. But a subset of these Roman flies actually had a mutation in this gene called ABO, meaning that they couldn't make an ABO protein in this particular case. And with the lack of an ABO protein, that is the thing that led to the infertility in these flies. So Larry was a magnificent scientist, but sadly he passed away at a very early age. In this perspective that's written by his co-conspirator in Rome, Dan, 12 years after his death, Dan really calls Larry an original, not just as a scientist, but just as a person who exuded a lot of joie de vivre. And of course, Larry left behind many legacies, scientific and personal, but one of those legacies was the flies that he collected, because other scientists continued to use them and study them. And one such scientist was an Italian scientist named Maria. So this was kind of a homecoming for the flies, coming from Italy to Seattle and back again. So the question that Maria was really interested in 2001 was, what is the mechanism? So we know that having no ABO protein leads to infertility, but what exactly is going on in that black box right there? So here, I'm gonna go back to the microscopic detour, but talk a little bit more about the histones that I touched on earlier. So histones are to DNA what spool is to yarn or thread. So, if you knit or if you know people who knit, you know that you can't just throw your yarn around because things get tangled. And much in the same way, you can't just have a bunch of DNA. You have to wind it around histones. And also it compacts it so that it can actually fit into the nucleus of a cell. And histones also are the answer to Maria's question of what is the mechanism. Because it turns out that the ABO protein actually regulates the amount of histones that is being made. But in the absence of the ABO protein, you get an overproduction of histones. And there can, of course, be such a thing as too much of a good thing. And this is such a case. So when you have too many histones, that results in decrease in fertility. So in 2020, I followed in Larry and Maria's footsteps and picked up the studies on these flies. But before coming to Seattle, I, like Nicole mentioned, was an ecologist. So I did a lot of field work with things like bats, tuco-tucos, which are these subterranean rodents in South America. And also I spent a lot of time in museums looking at mammal teeth. So of course, it's probably natural that when I picked up Larry's flies, I'm interested in looking at them through this evolutionary lens. And the puzzle that really attracted me to these flies is this rapid evolution of ABO. So what I mean by that is over this long evolutionary time, the ABO protein between Drosophila and melanogaster is very different from the ABO protein of its very closely related species, Drosophila simulans. So this is the puzzle that drew me to Larry's flies. But before I talk to you a little bit more about this puzzle, I want to spend a little time talking about one of my favorite things in the world, which is this little funny-looking structure on this left called the phylogeny. And evolutionary biologists spend a lot of time thinking about this. So phylogeny is sometimes also called an evolutionary tree, and it's essentially just a structure that depicts the relationship among different living things and different species. So, they can get very complicated, like the figures on the left and the center. On the very right, you actually have a phylogeny from one of Charles Darwin's personal notebooks. So, of course, things get complicated, but I always think it's best to start out simple. So here's a very simple phylogeny showing the relationship between 3 different species. And the key to reading a phylogeny is to look at these things called the nodes. And nodes are really important because that's what depicts the relationship between the different species on the tree. So, for example, the node on the left depicts the common ancestors of species A and B, whereas the node on the right depicts the common ancestors of all 3 species in the tree. So if you know to look at the nodes, you can read any phylogeny, however complex or convoluted looking it might seem initially. And the other really cool thing about phylogenies that I love is that it's kind of like a 3D sculpture that you're viewing on a 2D plane. So you can actually twist the node. So it's almost as if you were looking at a sculpture, like doing a 360 walk around the sculpture. So this phylogeny that you see here is actually identical to the phylogeny on the right. And initially you might be like, oh, the species order are different, but we now know to look at the nodes, and the nodes tell you that, oh, species A and B are still closely related to each other in both these phylogenies. So don't read a phylogeny left to right, look at the nodes. And in a similar way, you can also, turn it around, make it circular, just like do whatever your aesthetics leads you to do, just don't mess up the nodes because that changes the meaning of the relationships. So phylogeny 101, but I want to talk to you a little bit more about what we do with these phylogenies. So I created a phylogeny of electronics, specifically portable devices. And you can probably, just by looking at the different things on here, see that the criteria that I use to group these devices is what is portable and what has to be tethered, like a landline or a fax machine. Pictures just for fun. And you might be thinking at this point, like, wait, why does she put that pager right there? Because yeah, sure, a pager is portable, but you can't talk on a pager, whereas you can talk on a landline. So shouldn't a pager actually be grouped with the fax machine? And that, if you have that question, that's a very valid question. Because again, the criteria that I used for this phylogeny was portable versus non-portable devices. But I could have also used, as a proxy for estimating relatedness, this criteria of voice capability, right? And this actually is very reflective of how phylogenies work in real life. So often what scientists do is construct the phylogeny given a set of fossil evidence. And that person might also construct the phylogeny given genetic evidence. And they find that the two actually don't totally match. And then so now it becomes a job to then look at the parts that don't match and then look closely at the parts that don't match, and then gather other different sorts of evidence to see, for this given circumstance, is the fossil evidence a better proxy, or is the genetic material a better proxy? And the other thing that I want to stress about phylogenies is that phylogenies are best estimations or guesses that's based on the current available evidence. So this is not truth with a T. So of course, we're gathering new evidence all the time. So hypotheses change as we get more evidence. So as we gather more evidence, our phylogenies can change. So case in point, after I made this phylogeny, I was doing a little digging around on Wikipedia, and I found that I was wrong because apparently the precursor to the fax machine was invented before the precursor to the landline. And all of the modern mobile electronic devices, including the pager, use the technology developed for landlines rather than for fax machines. So my Wikipedia sleuthing tells me that this is the correct phylogeny. But if I get more evidence that's to the contrary, I'll just change it. So now that we've gone over electronics phylogeny, let's go back to our Drosophila phylogeny. So this is kind of a beast-web phylogeny showing lots of different Drosophila species, but we are just going to take a look at two of our favorites, Drosophila melanogaster and its friend simulans. So I'm going to bring you back to this puzzle that I'm interested in, this rapid evolution of abo. So by what I mean by rapid evolution of abo is that the abo protein within these two species, closely related species, is changing faster than expected over evolutionary time. And this is really puzzling because abo has this very essential function in flies. I'll just remind you that in melanogaster, abo controls the amount of histones that are being made, and that's important for fertility because if you get rid of this, too many histones and lots of babies die, so that's obviously not a great thing. So it's really this paradox of why is such an important thing changing? Because in biology, much like in electronics, essential things, so genes and proteins that are essential to an organism's function, tend to change slower over evolutionary time compared to things that are maybe more fibrous. So, for example, genes or pigments for hair color, that has more capacity to change because it doesn't super affect their health, right? And in a similar way, in electronics, your phone can be a bunch of different colors. That's just for fun. But every phone has to come with a charger because otherwise you can't charge your phones. And I think here is a bit of a paradox, and I think this might be the solution to this puzzle in the fruit flies, is if you're anything like me, that's very faint, but that's showing you different charging cords, essentially. And if you're anything like me, you're super frustrated because you have all these different charging cords. Like, I feel like every electronic comes with its own charging cord. And you're like, why can't they just make it the same? But that's how the way it is. So here you have an example of this very essential thing of like, my phone is useless without a charger. but the charger changes. And that's because the charging port on the phone has changed. So then the cord thing has to change to keep up, right? And I think that's essentially what's happening to the aboprotein in the fruit flies. So if you want, in this very faint picture, you can think of the aboproteins as the charging cords. And the thing that the aboprotein is binding to, which in this case is the histones, as the charging ports on the phone itself. So what I mean by that is what's happening at the molecular level is that the aboprotein binds to a very specific site on the histone gene. And this correct binding is really important. You can kind of think of it as like, sometimes I think I charge a phone, but then it turns out it wasn't like inserted all the way, and it's like my alarm doesn't go off in the morning or something, right? So it's very similar. Correct binding is very important here. So what could be happening in these fruit flies is that this particular portion of the histone gene that abo is binding to might be changing in the simulants compared to melanogaster. And if that's the case, then the abo for simulants also has to change to keep up, to be able to provide proper binding. So this is essentially, the hypothesis that I have. And in order to test this hypothesis, I'm doing something called a gene swap experiment. And here, what I'm asking is, can the aboprotein from these simulants bind correctly to the histone from the melanogaster? Another way to think about it is a phone charging experiment. So I know that this, lightning cable works with my iPhone and my friend's got an Android. We know that those things can charge. But what if I switch the cables? Can I charge my iPhone with my friend's Android charger? It's essentially what I'm doing with the fruit flies. So here you have the wild-type melanogaster flies, which you can think of as Apple products, if it helps. So you have the melanogaster flies that's got the melanogaster abo gene. It makes the melanogaster abo protein. But I've essentially engineered a set of melanogaster flies that now has the abo gene from simulants and makes the simulants abo protein. So if I were doing, the charger swap experiment that I was talking about of charging my phone with my friend's Android charger, the output that I'm measuring is my phone charged? In this case, the output that I'm measuring is, are the flies fertile? Because I'll remind you that in the melanogaster wild type, if you have the correct aboprotein, you get happy fertility and happy flies. But if you take away the aboprotein, so this is equivalent to essentially not charging your phone isn't charged, there's decreased fertility, we're not happy. And this is the case that I'm really interested in. So instead of not charging it, what if I swap its native protein with something that is slightly different? Can I still charge the phone, or is the fertility restored? So all of the stories that I've told you about fall under these are little volumes in this larger library of biology. And you might be thinking that, I hope you enjoy them because, I love flies, but, you might be thinking, these are very small stories on its own. Maybe you're even thinking this is really frivolous, you're not curing cancer, like, what are you doing? But with biology, like with all other sciences, in order for people to do things like make vaccines or create treatments for cancer, you first need some sort of baseline knowledge. So the scientists who are working on cancer research, who are working on COVID vaccines, they really rely on these individual stories and knowledge in this metaphorical library for them to be able to do their work. So really, basic sciences like the ones that I presented today, they really are the building blocks for the scientific and the medical advances that allow us to lead long and healthy lives. Thanks.
There is a Q&A session at the end for everyone, yes. So there will be time for questions, but we have 2 more speakers first. So our next speaker is Lucy Bowser, who is a graduate student in the School of Marine and Environmental Affairs at the University of Washington. She works on a project that highlights how fishermen protect the diversity of marine life and make up a crucial component of maintaining healthy oceans and a healthy planet. So please welcome Lucy. Thanks, Nicole. Switching.
Cool. Awesome. I'm really excited to share with you tonight a new way to protect our ocean community. Here we go. So, to start, we're going to move away from fruit flies for a moment, and we're going to the ocean. And so I invite you all to think of an ocean animal, just any ocean animal you'd like. Hopefully one pops up into your head. And this is where I'm going to practice my psychic abilities. Maybe some of you thought of our charismatic megafauna, like our whales, our sharks, or sea lions, while others of you might have pictured potential seafood, like tuna, shrimp, or crab. Or if any of you are ocean nerds like myself, maybe you thought of our lovely sea slug or a nudibranch, which is my favorite ocean creature. Regardless of what you visualized in your mind, there's a vast number of marine species that you could have pictured. And this vast number of marine species represents marine biodiversity. Now, unfortunately, marine biodiversity is declining. And there's numerous reasons for that, including climate change, extraction, pollution, and fishing. And I have fishing here that's highlighted a little bit bigger because that's what I focus on. I focus on fishing and how it relates to marine biodiversity because fishing is a vital resource for many people around the world as it provides foods, livelihoods, and culture. And I don't do this alone. I actually work with two other UW graduate or master's students in a capstone project where we aim to find this kind of balance between protecting marine biodiversity and also ensuring that people can still fish. Now, to do this, we can't do it by ourselves. We've partnered with the FAO, or the Food and Agriculture Organization. This is a very large governmental international organization, but essentially I like to call them the United Nations of Fishing. And with the help of FAO, or more, we're helping the FAO, we work with two other main groups. We have our conservationists, or those people who've dedicated their lives to protecting marine biodiversity and preventing its further decline. And we also work with fishermen, those who are actually out on the water day in and day out catching those fish for either themselves, their communities, or their nations at large. And so what we do with the FAO is we actually aim to bridge the gap between these two groups and have them kind of come together to address this problem of declining marine biodiversity and also protecting fishing. But before I get into how we go about doing that, first it's important to understand why marine biodiversity is crucial in the ocean in the first place. And to understand this, let's think of first a human community that's filled with people who have different roles. For example, we might have a stay-at-home mom, we might have a grocery store worker, and we might have a teacher. And through these roles, these people have relationships with each other. The mom might drop her child off with the teacher during the day and then go buy her groceries from the grocery store worker. And with these relationships, we have a coherent, cohesive community that functions pretty well. Same thing in the ocean. We have our ocean community, which is filled of marine species, and these marine species also have relationships with each other via food webs, or how marine species might eat one another. And similarly with these food webs, they support healthy ecosystems like coral reefs or kelp forests. And people care about healthy ecosystems because healthy ecosystems can provide us with things like food, support our economies, and then also provide us with some pretty cool tourist experiences. Now, if we go back to our human community, if we start removing people with their respective roles, for example, if we don't have a teacher or grocery store worker, then we start breaking the relationships they have with each other. Now, all of a sudden, our stay-at-home mom doesn't have anyone to drop her child off during the day with and doesn't have anyone to go get groceries from. And with the breakage of these relationships, then we start breaking down the function of the community at large. Same thing in the ocean. If you start removing too many marine species, you start breaking the relationships that they have with each other, and then ultimately the health of the ecosystems start to go down, and then all the benefits that people get from those ecosystems become threatened. And so this is a pretty big problem that our conservationists are trying to tackle. And in 2011, our conservationists said, hey, let's protect 10% of the ocean by 2020. Now, this 10% refers to certain areas or certain percentage of the ocean that is ultimately going to be protected from harmful human activity, which includes fishing. And the thought there is if you protect certain areas of the ocean from these harmful human activities, then ultimately you're protecting the biodiversity that's also in those areas. And so what did these conservationists do to try to achieve this 10%? Well, they relied on a strategy which I will be calling the red box strategy. Now, this red box strategy identifies geographical areas in the ocean, draws boundaries around these areas, and then, as I mentioned, prevents harmful human activities from occurring within those boundaries. And these areas on the map here, they're like reddish enough. You notice that they're spattered all across the world. And if your geography is good, then maybe you'll notice that some of these reddish areas are in places that are known for having high amounts of biodiversity, such as off of Australia, the Great Barrier Reef, and then off of Hawaii too, those coral reef ecosystems. And if we zoom in to one of these red box strategies a little bit closer, what does that look like? Again, like I mentioned, it requires the identification of like hotspots from marine biodiversity, and then pretty much draws a hypothetical red box around those hotspots, and then again regulates the human activity that occurs within them. And for these red boxes, that generally means prevention of humans inside those red boxes. Now, if you notice something from the schematic, we're missing somebody from this picture. As I mentioned before, our group not only thinks about marine conservation or marine protection, but we also think about the fishermen. And so with this red box strategy, where do our fishermen fit in? They fit in outside of the red box. You can think of that red box meaning fishermen, stop, don't go there. And this is important because in those areas, fishermen are prevented from fishing, and that can be potentially harmful to the fishermen, especially if their livelihoods and their culture are tied to those fishing practices. Nevertheless, our conservationists were relying on this red box strategy, and so 2020 came and went, and was this 10% achieved? It was not. However, 2030 is on the horizon, and so our conservationists now are saying, let's bump it up, let's protect 30% of the oceans by 2030. This is called the 30 by 30 goal, has a nice ring to it. And the conservationists recognized that with the 10% goal, the red box strategies were not enough. And so now they're thinking about another way to conserve or protect marine biodiversity. They're looking elsewhere. And where are they looking? They're looking to the fishermen, right? As I mentioned previously, marine species comprise the ocean community, but the fishermen do as well, as they are out there every single day catching the fish, and they're out on the water. And so the fishermen need to be included in these global biodiversity conversations because they are the ones who are most impacted by the decisions that our conservationists make, like through the red box strategy. Also, it was evident that with the failure to achieve that 10%, the marine protection and marine conservation is really a team effort. The conservationists were not able to achieve that goal alone, and so by incorporating the fishermen with trying to achieve these goals, now we can start to try to expand the efforts to more people. And then also, fishermen, like I mentioned, a lot of them have their cultural practices tied to fishing, and so it's important that the conservationists also recognize and respect that as well. And by doing that, by incorporating fishermen with these global biodiversity conservation efforts, then now we're addressing the holistic ocean community so that we can begin to find management strategies that are going to be beneficial for the marine species and then also for the people. And so when we're looking at our fishermen are placed in a tough spot because the ocean biodiversity is declining, and yet there's an ever-growing demand for ocean resources. And some fishermen recognize this kind of imbalance, and they have actually decided to use fishing practices that are less harmful to the marine environment. And fishermen that are using these less harmful fishing practices fish in areas that I will denote with a green box. Within this green box, our fishermen think about the fish, but they also think about the habitat that the fish live in, and then they think about the other species that those fish are related to or interact with. And by doing this green box strategy, these fishermen are already taking a more holistic approach to fishing by incorporating how fishing might impact the broader ecosystems. And so, what might an example of this green box look like? Here we have a hypothetical example starring Fish Boy, someone you or someone you know may or may not have swiped yes or no on, holding two halibut, something a lot of people fish for. And our halibut, denoted here by this little cartoon icon, they live at the bottom of the ocean, along with other marine species, and there's two main ways to fish for halibut. You can use a long line, what you can imagine is a really long fishing line that extends to the bottom of the ocean, and it has hooks on it intended to catch halibut. And then you can also catch them via bottom trawling, or instead of a long fishing line, you have a massive net that gets dragged along the bottom of the ocean, and then it scoops everything up. Of these two fishing practices, longline fishing actually protects some of those other marine species that live at the bottom because they're intended to selectively catch the halibut, versus the bottom trawling does not discriminate between those marine species that live at the bottom and is ultimately more destructive to that habitat. And so if we look at our green box again, that area where these halibut fishermen are fishing, within the green box we have that bottom habitat that include the halibut and other species, and we have longline fishing that is allowed within this green box boundary, and bottom trawling is not. And so while bottom trawling can occur in other areas of the ocean, within this green box, bottom trawling is prevented, and ultimately some of that bottom habitat is actually being protected. And so this green box differs from that previous red box strategy because fishermen who use longline fishing practices are allowed to fish within this boundary, but then also that more destructive form of fishing, the bottom trawling, is being prevented. And so this green box strategy highlights a fishing practice that already is being done around the world in certain parts, and then it has benefits to marine biodiversity. It also reflects better ocean management because it allows people to still fish, it acknowledges that need, but then also acknowledges that marine biodiversity needs to be protected as well. And so, when our conservationists are looking for other strategies to go about protecting marine biodiversity, these green boxes are a pretty good place to start. And so where do these green boxes exist? They also exist all over the world. For simplicity reasons, I kind of roughly drew in where these green boxes would be for these given nations. But what's really important to note is that these green boxes are separate geographical areas from those red boxes. They don't overlap. And again, the rules within the green boxes are different from the rules within the red boxes. And so the green boxes have benefits to marine biodiversity, as I mentioned in my example, because of the prioritization of those less harmful fishing practices. However, these green boxes, and ultimately fishermen, are not officially recognized for their contributions to marine biodiversity protection. And so to change that, we have a new strategy that has recently emerged called OECMs. OECMs were made by our conservationists to include the fishermen in these conservation efforts and stand for other effective area-based conservation measures. A mouthful, I know. But what you can really think of OECMs as is they're a policy tool that will allow the 30 by 30 to become a reality. And if we go to how do we achieve that 30% ocean protection, on the left here, if we don't incorporate these green boxes, then we'll need to implement more red box strategies in order to try to achieve this 30%, which, as I mentioned, could potentially be harmful to the fishermen and communities that would be prevented from fishing in those areas. With OECMs, we can now formally recognize those green boxes and their contributions to biodiversity protection and ultimately allow them to count towards that 30%. It might not be a 50-50 split, but 30% can now be achieved with less disruption to fishermen and then also, again, push for better ocean management that balances the needs between protecting marine life and then also allowing fishermen to fish. OECMs don't come without their challenges, and the main challenge right now is we have a green box where fishermen are fishing, but then how do we actually know that biodiversity is being protected within that green box? This question of knowing whether or not biodiversity is being protected is a question that confuses many people, because biodiversity itself is something that is tricky to measure, monitor, and evaluate. And then it can be even more confusing when you add fishing on top of that. So, OECMs, instead of standing for that really long thing, have been coined other extremely complicated methods. The added bonus to this, too, is that OECMs are so new. They've only been around since 2018. There's really not any good examples that we can point to illustrate what the process of designating a green box as an OECM would actually look like. And so if I go back to my capstone group and our work with the FAO, what have we been trying to do to help maybe find solutions to answer this question? We've been holding informative workshops where we actually bring people together to talk about these green boxes and whether or not they would in fact count as an OECM, and then ultimately towards that 30%. And one workshop in particular began to answer the question, what do we measure or what do we actually look for that's within these green boxes that will tell us something about the status of the biodiversity? And so this expert workshop brought together people from both groups, our conservationists and our fishermen, and it began to discuss these things called indicators, or what we can measure to tell us about biodiversity. And so with our green boxes, these indicators are going to be something that's found within the green boxes. Maybe it's measured, maybe it's counted. That will tell us either, yes, biodiversity is being protected, or no, it's not being protected. And from this workshop, we came up with preliminary indicators. For example, you could count the number of non-target fish. If you go back to that halibut example, that would be counting not the halibut themselves, but an associated species of fish that lives down at the bottom with the halibut. You could also count the number of baby fish or the number of baby halibut. You can count the number of adult fish or adult halibut, or you could count the number of threats reduced, right? For that example, one threat was reduced. We're not bottom trawling in that area. And so these indicators and this discussion from this workshop really is just the start of this work, because indicators, as you could imagine, would be influenced by people's backgrounds and their interest. And so coming together and trying to find a very definitive list of indicators that everybody agrees on is quite challenging. And so we might have come up with some preliminary indicators, but overall the big success was that dialogue between the conservationists and the fishermen have begun. And this is a benefit to this process with green boxes and OECMs, because we're incorporating those fishermen right with these discussions, and that was lacking with the previous red box strategy. And so if we're thinking about green boxes and OECMs in the future, what's going to happen? We're going to continue these conversations. Again, it doesn't end with one discussion. It will be many down the line between our conservationists and our fishermen. This work with the indicators will also continue, as the indicators are those really important things that will tell us whether or not green boxes are OECMs. And so we're going to keep working on that. And then that question of, will 30 by 30 be achieved? We're not sure. Maybe it will, maybe it won't. But the bigger thing that we're pushing for with these green boxes and OECMs is better ocean management. Again, trying to find that balance and of strategies and management that will not only protect marine biodiversity, but also protect people's livelihoods, those of the fishermen. Thank you.
I'm not trained on using this computer. Let's see. Cool. Okay. Thank you, Lucy, for an awesome talk. Our final speaker for the night is Elizabeth Bonner, who is a PhD student studying age-related blood cancers at Fred Hutchinson Cancer Center through the University of Washington's Molecular and Cellular Biology program. She studies the most frequent mutation found in a group of age-related blood cancers to understand how this mutation disrupts the production of blood cells. So please welcome Elizabeth. Sorry, I left it on the big screen.
Oh, I was like, where's my mouse? There I am. There's that. All right. So good evening, everyone. Thank you so much for sticking around for my talk. So today I'm going to be talking to you about my thesis work, which is looking at better understanding blood cancer, how good blood goes bad with age. And so before I get into kind of like the nuts and bolts of my thesis work, I'd like to share a fact with you, one that keeps me coming into lab day in and day out, despite how difficult science can be. And that is the fact that your cells are amazing and also a little scary. So what do I mean by your cells are amazing? As Risa mentioned, each cell in your body is the smallest unit of life. And it's capable of performing really complicated tasks thanks to its microscopic and poetically intricate makeup. Each cell in your body contains factories capable of making every tool that it could ever possibly need to do its activity and to survive. Each cell also contains processing plants capable of taking nutrients from the environment, the food that you eat, and turning that into energy to do this work. Each cell is capable of responding and listening to its environment, and each cell possesses this really complex structure which allows it to move and change shape. And all of these complicated tasks are coordinated thanks to this vast library contained in a cell's core. Contained in this library is information needed to make every tool any cell could ever possibly need. It's also information which makes you uniquely you. So when we think about the cells of our body, the cells in our teeth, which make them hard, the cells in our eyes, which respond to light, our heart cells, which allow it to beat, or our brain cells, which create our thoughts and dreams. We could think of each of these cells as being unique and different to their location and function. However, each of these cells evolved from one group of cells which came into existence upon our conception, and those are our stem cells. So this is absolutely amazing to me. All of these diverse cells, which have these diverse and unique functions, all evolved from one population of cells. So the field of stem cells is huge. You have stem cells all over your body. You have stem cells in your gut, and you have stem cells in your skin, and you even have stem cells in your brain. So rather than talk to you about this really broad field today, I'm going to talk to you about the first stem cell ever discovered, and that is your blood stem cell. Blood stem cells give rise to all the cells of our peripheral blood, cells responsible for immunity, carrying oxygen, and blood clotting. And the way our blood stem cells give rise to all of these diverse cells is through a process called hematopoiesis. So what is hematopoiesis and how does this work? So I want you to imagine that you have a set of identical triplets, and your identical triplets live at home with you and they go to school. And throughout the course of their lives, as they travel back and forth from school to home, they're each going to experience unique and different things. One of your triplets may see a fireman rushing into a burning building and just be super inspired by their heroism. Another one of your triplets might hear a piece of music and just be absolutely moved by the melody. And your final triplet may just really dig staying at home and playing video games. Well, each of these unique experiences are going to shape who your triplets are and how they interact with their environment and at school. They may join different friend groups, or they may take different classes which directly speak to their new and budding interests. Eventually, your triplets are going to grow up, they're going to finish school, and they may even go off to college. Eventually, they're going to leave the house, and maybe they're going to move all across the country to complete their training and to pursue unique careers, contributing to society in their own way. Well, this process of your twins growing into your triplets growing into unique individuals is akin to the process of hematopoiesis. But instead of talking about human babies, we're now talking about your blood stem cells, or BSCs. Blood stem cells call their home the bone marrow, which is located on the inside of our bones. And for education, our blood stem cells listen to different environmental cues. So just like with your triplets, our blood stem cells will encounter different environmental cues within the bone marrow. And as a result, they'll start to mature in different ways. And as a result of this education that they receive, eventually your blood stem cells are going to leave their home in the bone marrow, and they're going to travel to different parts of your body where they're going to perform their various functions, such as immunity to fight infections, blood clotting to help stop bleeding, and oxygen transport to help carry oxygen throughout the body. So our blood stem cells are capable of performing this very complicated task thanks to information carried in their DNA. So how does our DNA coordinate this? Well, you can think of DNA like an instruction manual. And in this instruction manual, there are instructions to make every tool you could ever possibly need to do any kind of job. So let's say that the job you need to do requires a hammer. Maybe you're building a bookshelf or something. Well, you would turn to the section in your instruction manual that tells you how to make hammers. You would find the instructions for the specific hammer that you need to make, and you'd build it. Well, this is the way that our blood stem cells make their tools. You can think of the DNA like your instruction manual. And just like you, your blood cells only need specific tools to do specific jobs. So those specific instructions for those specific tools are encoded in a gene, a piece of our DNA. And then when our cells need to do these specific jobs, they turn to that gene and turn it on to make a protein, or the tools that cells use to do work. So this is pretty amazing. Our blood stem cells respond to environmental cues to make our peripheral blood cells by turning to their instruction manual, DNA, and taking genes and making proteins, the tools which they need to do work. Now, the process of going from a blood stem cell to a blood cell is a big job. And for a big job, you're going to need a lot of tools. So instead of building a bookshelf, now let's say you're doing a really big job. Maybe you want to turn your house into a McMansion. Well, you're going to need a lot more than one hammer to do this job. And rather than building one hammer by hand, at a time, it would probably be more efficient to use some kind of machinery or factory to build all these hammers that you need. Well, unlike you, factories and machines aren't very good at reading human language. So you would probably want to use some kind of code that the machines can read to really ramp up their production. Well, this is exactly what our cells have to do to mass produce their tools, proteins. But rather than computer code, our cells use RNA, which allows our cells to ramp up protein production. So in our instruction manual, there's going to be a lot of information that the machines aren't necessarily going to be able to read. Well, the same is true for our genes. Within our genes, there is information which may be important for the gene, but isn't necessarily important for protein production. So in order for our genes to mass produce proteins, this information needs to be removed. And this is a process called RNA splicing, where this unimportant protein information is removed, which then allows our cells to ramp up protein production. So this is super amazing to me, that our blood stem cells do this really big job of listening to environmental cues to make cells critical for our daily life by turning to their DNA, finding the right gene, turning that into RNA, and mass-producing proteins to do work. So in the beginning, I told you that our cells can also be a little bit scary. Well, our blood stem cells, which produce cells critical for daily life, can stop listening to environmental cues as we age. And this means that they're no longer efficiently producing blood cells. This occurs because as we age, our blood stem cells can acquire mutations in genes. Every time our blood stem cells reproduce and grow, they must copy their DNA. So this is akin to making a photocopy of our instruction manual. With each successive copy, the quality is reduced, it becomes harder to read, and errors can arise. Imagine trying to build your hammer from a page that's been copied 50 or 100 times. Some information may become lost or damaged, which means that you're now building a tool with either no function or some unknown function. Well, this is what can happen to our genes as we age. Errors can occur, which causes our cells to produce proteins of either some unknown function or the protein can be lost, meaning that it no longer does the work that it's supposed to do. And when this occurs in our blood stem cells, this can lead to the development of blood cancers. So for my work, I study one specific type of blood cancer known as myelodysplastic syndrome, or MDS. So MDS occurs because as an individual ages, one of their blood stem cells may acquire a mutation. This mutation doesn't kill the cell, so the cell continues to grow, but it may affect its function and ability to produce blood cells. As time goes on, the number of this one blood cell continues to grow within the bone marrow, and eventually the number becomes large enough that blood cell production is affected. This can lead to a depreciable quality of life for individuals who experience myelodysplastic syndrome. Individuals with MDS no longer produce cells important for fighting infections, which means they can get sick easily. They also no longer produce cells important for blood clotting, which means they can bleed easier and longer, which makes even small bumps and bangs very dangerous. Additionally, they no longer efficiently produce cells important for carrying oxygen, which means that they can tire easily and increases the risk of slip and falls. And in addition to these issues, if the blood stem cell acquires a mutation which causes it to grow and reproduce rapidly, that blood stem cell can overtake healthy cells within the bone marrow and lead to more serious forms of blood cancer such as leukemia. So scientists have spent the last decade trying to understand which mutations lead to the development of myelodysplastic syndrome. And what I'm about to show you in this graph is the frequency of certain types of mutations which they have found. Some of the most common mutations occur in things like DNA marks and DNA accessibility, but rather than delving into kind of what all these mutations mean, you can largely think of these mutations as a group of genes responsible for accurately reading and copying DNA. Most interestingly, what scientists have found is that the most frequent gene mutation which occurs in myelodysplastic syndrome are mutations in genes involved in splicing. So what happens when these splicing mutations what happens when these splicing genes are mutated? As I mentioned before, splicing is the process which takes unneeded information in an RNA and removes it such that a cell can ramp up protein production. Well, when splicing genes become mutated, this process no longer works effectively. RNAs can have whole chunks of important information or just tiny bits of important information removed. Or they can have some of that unimportant information left behind, which means that cells can mass produce either bad proteins or they can lose these proteins. So, it's cut off. So this is a little scary. Splicing mutations can cause our blood stem cells to no longer respond to environmental cues, leading to a loss of cells which are critical for our daily life. And this can happen due to the fact that even if a gene itself is not mutated, it no longer effectively produces functional proteins because the RNA, which is responsible for the cell producing those proteins, is no longer processed correctly. This is made even scarier when we consider the therapeutics available for myelodysplastic syndrome. There are no targeted treatments. The only therapies available for most individuals are regular blood transfusions to replace oxygen transporting cells or blood clotting cells. But this is costly, it's uncomfortable, it's time consuming, and it doesn't directly address the issue with the blood stem cells. The only potentially curative therapy is what's known as a bone marrow transplant, where a patient's blood producing cells are eliminated using either chemotherapy or radiation, and then the blood stem cells are replaced with donor cells. This is a very dangerous procedure. It's highly invasive, and it doesn't necessarily always fix the problem. Additionally, most individuals are not eligible for bone marrow transplant due to other age-related illnesses, such as heart disease, or due to the lack of donor availability. So, the work being done by me and my lab seeks to address these shortcomings. Can we develop better therapeutics for individuals with myelodysplastic syndromes? So one of the ways that I'm looking to find therapeutic targets is to use high-powered computers. What I do is I can collect blood stem cells from healthy individuals as well as individuals with splicing mutations in myelodysplastic syndrome and extract RNA and then ask what's different. What RNAs are lost? What RNAs are misspliced? So what I have found is that splicing mutations can disrupt hundreds of proteins by both reducing the amount of an RNA and by missplicing it. On the graph I'm showing you is one of my analyses. Each dot represents a different RNA, and the color of that dot indicates how that RNA is misspliced. The further to the left a dot is, the less of that RNA there is in a cell, and the further to the top that dot is, the more misspliced that RNA is. So, as you can clearly see, splicing mutations disrupt hundreds of proteins. Sorry. Often in cancer, a single gene mutation will only disrupt the function of a single protein. However, in splicing mutations, one mutation is capable of disrupting the function of hundreds of proteins, which makes understanding these mutations even more difficult. So over the last decade, many scientists have thought that if we can identify and characterize these missplicing events, we may be able to find treatments. My thesis mentor found that this missplicing event leads to increased inflammation in blood stem cells, which can disrupt their function. Another group at Fred Hutch found that this missplicing event causes solid tumors to grow faster because splicing mutations can also be found in some solid tumor types as well. And another group found that missplicing of this event leads to poor oxygen transport by red blood cells. However, despite all of this work, we still don't have an understanding of how splicing mutations lead to the development of myelodysplastic syndromes. So, unfortunately, I'm going to have to leave you on a little bit of a cliffhanger. Where it stands, more work is still needed. But have no fear. Many scientists, such as my own lab, are working diligently on this. For my work, I am continuing to test misspliced RNAs. This is an uphill battle, but I am in for the climb. Additionally, I'm also using new technologies to see if we haven't missed some missplicing events. It's my belief that if we can find the missplicing events which lead to the development of MDS, that we may be able to find new therapies or utilize existing therapies to better treat these patients. So if you wish to learn more about the work that is being done by my lab, I would highly recommend that you come to my lab mate Rasika's talk on May 15th. I'm not going to give any spoilers, but her work is super cool, and so I highly recommend coming to check it out. And with that, I will say thank you for your attention, and I will pass the mic back to Nicole.
Yeah, so if you have any questions, feel free to come up to the mic, and we're here.
I have a question for the first speaker.
Yeah.
I love your topic on the libraries, one of my favorite places. I'm currently trying to struggle through understanding traditional taxonomy and the new cladistics, and you didn't talk about clades in your talk. Where does that fit in to the library?
So when you say clades,
Well, when I look at taxonomy of marine animals, for instance, a lot of times they'll say this animal in classical Linnaeus
clade.
used to be in this family, but now using cladistics, it's moved over to a different family.
Sure, yeah. So the term clade it refers to a grouping. So, you know, those orange boxes that I had in my talk, those are referred to. One group is called a clade. And cladistics is a certain, let me just make sure I'm getting this right. It's like a school of thought, or like you mentioned, it's like a method of trying to resolve these phylogenies. And there's a lot of people get unbelievably, I think, heated about what is the best method. You know, to be honest, I don't have any horse in the race, but I have heard that these arguments can like end in people yelling across the rooms and stuff. But so to go back to your question, so I think maybe I'm not answering your question directly, but I think it.
Trying to learn more about
Yeah, I think it does kind of touch, right, I think it does touch on the fact that, all of phylogeny, what we're trying to do is understand what is really the correct relationship between these living things. And then there's just simply different methods to arrive at that answer. And I think sometimes certain methods turn out to be wrong. Other times it's not that one method is wrong, but it's not right for this particular circumstance. And so it's just one method of doing phylogeny.
follow-up question.
Yeah.
So I mean, originally people used to call whales fish, but because they're
Yeah.
in the ocean. So as
Right.
we move on, they
Sure.
get more refined and more refined.
Sure.
Is it correct if I assume that cladistics is based on DNA analysis of the animal, or is that an incorrect assumption?
I would have to check my notes. I don't want to say something misleading, so I don't know off the top of my head. But yeah, different analysis can use, like fossil evidence or DNA evidence. But I think now what a lot of people recognize is that it's not that one is wrong, but oftentimes these things can be complementary to one another. So essentially, my take on it, this is just my two cents on it, is that the more lines of evidence that we can gather, the more resolved a phylogeny that we can get.
Thank you.
As much as we all love the Roman farmers market, is there anything wrong with, good old American fruit flies? Is there a reason they had to use imported Italian fruit flies? Extra virgin?
It was a very
imported or-
political decision. No, it just certainly happened that there was a lot of fruit fly research going on near Rome, Italy at the time. And there were previous scientists who were like, we collected some really cool flies here. So the American scientists, I think, had a pretty fun sabbatical. They went with their families to Rome, and they took road trips and different wineries and stuff. So yeah, so there's a lot of fruit fly collection going on all over the world. There is a lot of flies, and it's really cool because the fruit fly species that you might find on your counter and kitchen counter in Seattle is actually slightly different, or subspecies compared to fruit flies that you might find in Arizona, for example. So there's a lot of fruit fly diversity here.
Does anybody else have a question? Feel free to come up.
A little bit. Okay. So the first question I have is for the talk on the ocean community, and I'm curious about. So you're incorporating like fishermen and the conservationists, and then, but I'm also curious about how you're. Maybe I don't know if you've thought about like how do you incorporate the communities, like for example near the green boxes, because they also have a say. They should have a say in the policy that goes through. So I'm curious what's happening in that.
Yeah, what a great question. Something that if you look into OECMs and kind of more of the actual rules of them, if you will, one of the things that's pretty cool about them that the red boxes don't do is that they actually acknowledge that communities have, especially indigenous nations as well, have their in sovereignty, right, and what's the best word? Sovereignty and the inherent right to rule or manage the areas that they live in, and so that's actually one of the trickier aspects because you can write that in there, right? Say communities should be able to locally manage their own areas for the benefits of the greater biodiversity or whatever, but actually allowing them to do that is a little bit trickier than having it simply be written. But it is something that governments, because the policy tools that I'm talking about are coming from the top down, they're coming from national governments, and then that gets translated to the local government, right? And so with that kind of cross-disciplinary work of the different scales of governance, I think they're acknowledging, right, that it needs to not only come from top-down management, but also the bottom-up as well. But again, just we're working on trying to engaging the local communities, making sure that their needs are met too. But there's still improvement that can obviously be done as well. But we're shifting in that direction, and it's becoming more and more prioritized, I think.
Great, thank you. And then another question I have is for the talk on the blood stem cells. So one approach is to look at the different therapeutic targets, but is there any previous work that's shown that it's possible to maybe regenerate this pool of blood stem cells? Is that possible?
That is a great question. So our blood stem cells kind of always are there. So you could regenerate those blood stem cells, but there's a finite amount of space within your bones, right? So like you're still going to have like the deficient blood stem cells, and so long as those are there, you're still going to have deficits in your ability to produce peripheral blood cells. The other kind of issue with that approach is that we are really bad at working with stem cells. Like they're really complicated little guys that don't like to behave. So everybody's looking for that next, like, can we take out a healthy, any stem cell, like pick a stem cell, can we grow it in addition, can we put it back in you to like fix you? It's a huge field of work, and it is going very slow. So hopefully, maybe someday, but yeah, not yet.
Thank you.
Mm-hmm.
This is a biodiversity question. The polite way to ask it is, how do you measure biodiversity? And the crude way to answer it is, if I go in and kill half the kelp and half the otter and half the urchins, and do I still have the same biodiversity?
That is a great question. And you're right.
Looking at those bottom trawlers. For
Yes, and trawling is actually one of the ways that scientists have traditionally used to actually count the things in the ocean, is they take the things out of the ocean to count the things. And I think that's really interesting. We're seeing a shift now with emerging technologies. I don't know, eDNA or environmental DNA is one of those technologies that's still kind of up and coming, but many people hope that you can just take a water sample and living things shed DNA, and you can get a snapshot of things that were in that time and place in the water without having to remove those species from the water too. That's the example I have off the top of my head. I think there's other ways, right, to actually measure, count biodiversity that people are trying to do that are less harmful to the actual animals themselves. So we're kind of, it's again, very like slow transition away, but we are working towards that too, like we, meaning like scientists in the ocean, not myself per se, but yeah, so it goes along with emerging technology too.
I have a question about that article in Science Magazine that was retracted in 2021, primarily because, wasn't it a UW's fisheries expert that testified that there was, that from what he could see, there was no decline in fisheries, and the MPAs were supposed to increase the fishery by 5%, and of course they retracted it. So I'm a UW graduate, but I was pretty amazed that UW got this involved, and certainly in the fisheries department. You know, I wonder about his, I read his articles, and it seemed like he was on track with what he was measuring. I mean, I'm just a scuba diver, but I've seen marine diversity and quantity and quality of the fisheries all over the world decrease dramatically in my lifetime. So what's going on in the fisheries department?
You know, also great question. I'm not actually in the fisheries department. I'm in a department that is more so ocean policy at kind of broad scale. And so the fisheries department is actually, we're SMEA, they're SAFS, the School of Aquatic and Fishery Science. I don't know what's going on over there. but University of Washington is...
Accurate to me.
Yeah.
So what was he measuring that's different than what I'm seeing?
I think that goes back to kind of the point, I think what you're talking about with like clades is, right, as I mentioned, that there's different ways to measure biodiversity, and depending on what way you pick, you might get a different image of biodiversity as a whole. And so it also kind of comes with just like new information, new methods to measure biodiversity. The politics of the department might be a little bit different and something that I cannot speak to. But yeah, just that's kind of the way that science works. It's not just like we know it and that's it. It's a process. And so sometimes we might know some things. I had a professor who was like, yeah, I thought this was the best model to look at ocean equity. Turns out he like just published a paper and he's like, maybe it wasn't. So it's a process where getting new information and having different perspectives on the same thing can change kind of maybe your outlook on a paper that you wrote, for example. Yeah.
I have another different question if no one's standing in line.
It sounds like, did you have a question? I'm sorry. I'm sure we'll have more time to come back to it.
This is a question about the stem cells, or the blood cells. With the coming of AI, how is that affecting research in the complexity and understanding of the stem cells? And you know, for some things, some things are very difficult for humans. They're amazingly easy for computers, and I just wanted to ask about that.
That's a great question, and it's super exciting. So with the advancement of new technologies, we now have the ability to look at a cell-by-cell basis at things like what's the DNA, what's the RNA, and a whole host of other things. And we have had something called machine learning for quite some time, where we can take a training data set or maybe some old data that's been like really well validated and feed it into machine learning, and it kind of spits out some kind of weird, it spits out these giant blobby maps that are kind of like, but with all of the new artificial intelligence coming, and with all of the new advancements in computing and machine learning, this information is becoming far more better understood. So I think ultimately we're still quite a ways away, because computers and programs are only as good as the people who program them, and there's still so much information that we need to understand for hematopoiesis or other single-cell data sets, but it's definitely, it's like a ball rolling downhill. The momentum becomes faster and faster, and I think our knowledge on things like hematopoiesis will expand ever quicker with the advancement of things like machine learning.
Can I ask a follow-up question?
Sure.
I've had the same experience in chess. It's like we are no longer programming the chess computer. It programs itself. And in the same way, you know, I wonder if maybe we're getting, we're getting in the way necessarily of the research. You know, in other words, the computer could program itself so that it's the only one that really understands what it's doing. And then in that case, it's only a question of whether you get the result you're looking for.
That is super fair. So that is absolutely fair, that because what we do is called like supervised clustering. So that's where we say, put all of the things that look similar here because this is what we've defined. So it's absolutely possible that the computers could far surpass our understanding, but where computers fall short is they don't have hands to test the results. So we can do all we want in what we call in silico in the computer, but code can be wrong, and your input could be wrong. Like you could just have really icky cells that are doing something really icky and they're not representative. So no matter what happens kind of in silico in the computer, you still need scientists like all of us to get in there and get your hands dirty and do the actual work. So we may be getting in the way of computers, but they can only do so much for us. In the end of the day, nothing that happens in a computer means anything until we can take the cell and manipulate it on a molecular level to make sure that what it's telling us is correct.
Thank you.
Yeah, no problem.
I have a question about ocean conservation. So I'm not particularly well versed in conservation or policy. Perhaps this is a bad take, but it seems to me on the surface like the way we go about ocean conservation versus conservation of areas on land is very different. So it seems like with land conservation, we kind of like rally around like these, charismatic fauna, things like lions and pandas. And it seems like to me the goal of that is to bring in more stakeholders into it. So kind of maybe like perhaps raise the interest in, for example, the American public in a place like, like sub-Saharan Africa where there's like lions and rhinos. With oceans, it seems like there's a very different approach. And so because of that, it seems like the group of stakeholders is very different. So in your talk, you're kind of talking about how the stakeholders primarily were just, the people living in the area and the people fishing the area. So do you think kind of this difference in sets of stakeholders is a benefit or a hindrance to the preservation of the oceans compared to land? conservation?
Great question. One of the things that's tricky in the ocean, because conservation efforts from land to ocean need to be different, water moves and land doesn't, and a lot of species in the ocean land on land too, but they move like miles, right? Like you have big migration events. And so it does kind of depend on if you decide to look at maybe like a kelp forest or something, like that's a habitat that's relatively fixed in that space, but you have that water that moves through that ecosystem. So if you're thinking of environmental kind of factors, right, that might change and be influenced by other things. But regarding the stakeholders, to your question, you can't really have like that one-to-one from conservation to land to the ocean because, right, the people are going to be different. And then also it kind of depends on that scale at which you're hoping to conserve like a certain area of the ocean. Many like cross boundaries of different nations. So you have like different governments that are coming in, or they have also like big international kind of like organizations, for example, that focus on the tuna, as tuna are highly migratory that spanned miles of the ocean. And so it kind of depends on what area you're looking at and the scale that it is at that dictate kind of who is involved. And I did simplify it down to like the fishermen that are in the area and local communities that would be fishing predominantly for their livelihoods, because those are generally the people that get overlooked when it comes to conservation in the ocean. Mainly, it's been those top-down kind of like big national governments or even international bodies saying like this is how we're going to conserve. And although there are other people that are affected by those decisions, they really haven't been incorporated into those conservation efforts. And so now what we're trying to focus on is, again, that kind of bottom-up approach, going to be like, hey, this community or maybe this nation in this area, right, we want to do this conservation effort, you're here, right, kind of what do you need in order for that to actually work, right, and be beneficial for them as well. I feel like I got sidetracked from your question. Was that, it's contextual. That's the short of it. Not a very satisfying answer. All right.
Oh, yeah, go ahead. This is probably, I think, our last question we need to wrap up around. Is that right?
Hi, I was wondering about the fruit flies. Is there any significant difference in like lifespan between the Roman market flies and DML? And what I'm getting at is, due to the errors in histone packaging, are there associated comorbidities?
Yeah, that's a really good question. So it seems like there isn't, although there is a hypothesis that's actually kind of maybe a little bit counterintuitive that I haven't tested. But so these histones, having an overproduction of histones actually might be bad in the reproductive stage, but it actually maybe could help an aging fly live a little bit longer. So that's a hypothesis that's out there. And because these lifespan assays take a long time, I have not attempted it yet, in parentheses, but that's a really interesting point. Yeah. And on the topic of flies, these are...
I was going to ask that.
Yeah, so I brought some, if anyone's interested, afterwards I can show you. These are the flies that we keep in lab. So we have these empty vials. So this is a food, this tends to be, this particular one is corn syrup, but you can also have it be molasses. It actually smells really great when it's being made because it's like high fructose corn syrup or whatever. Or I don't know if it is, but it smells like that. And then here you have a newer bottle where you have the adult flies and then the larvae are growing here. And this is a slightly older bottle where you can kind of see pupae that's emerging on the walls. So these guys are super cool.
All right, I feel like that's a really good place to end. So yeah, I mean, if you all have more questions for our speakers, I'm sure people will be around for a minute. So feel free to come ask them questions one-on-one. Otherwise, I just want to say thank you for joining us tonight. We want to invite you back for our next Engage Science Now event, which is on May 10th, starting at 5:30, also here in Town Hall. We'll have 3 more speakers from the class giving some awesome talks on their graduate research. If you want to learn more about Engage, you can either come chat with me after this. I'm happy to share and answer any questions. You can also visit our website, which is engage-science.space, or you can follow us on our social media. We need more followers, so please do. That is at Engage Science on either Instagram or Twitter. And if you happen to be a graduate student at the University of Washington and you're excited to learn more about science communication, you should consider applying for our course next year. The course runs in the winter quarter, so winter 2024, and applications normally open in November, and I'm happy to answer any questions if you have questions about that too. But let's thank our speakers one last time.